이번에는 Your Inner Fish, 몸의 비밀, An Evolution Story 3부작 중에서 1부, 2부는 했고 3부 Your Inner Monkey, 우리 안에 원숭이입니다. 인간의 몸에 새겨진 흔적을 조사해보면 다른 동물의 유산임을 알 수 있다. 다른 짐승과 구별되는 인간 고유의 것으로 보여지는 몇 가지 특징이 있는데, 첫 번째, 자유자재로 사용하는 손이 있고, 두 번째는 다양한 색깔을 구별하는 눈이 있고, 세 번째는 서서 두 발로 걷는 직립보행이 있고, 네 번째는 고도로 발달된 뇌가 있습니다. 이러한 손, 발, 눈, 뇌 등이 모든 물고기, 파충류, 원숭이 등의 다른 종에서 유래되었다는 거죠. 손에 관련된 유산이죠.
To see what I mean, imagine all life that has ever existed on a giant family tree. From the first microscopic life billions of years ago to all animals alive today. We didn't evolve from modern monkeys, but if you trace our ancestry back in time, eventually we reach a point where the human line and the lines of all primates meet. This is where our story begins, our common ancestor. The ancestor of every monkey, ape, and human alive today. So what did that common ancestor look like? And how has it shaped our own bodies? 1870. A surgeon from the Civil War has returned to his pre-war passion, hunting fossils from the Wild West. In Wyoming territory, he finds a jawbone he thinks might belong to something like a small raccoon. In fact, he's found a creature that lived some 50 million years ago and occupied a place in our family tree very close to the first primates. I've come to meet this creature called Netharctus, and a scientist who knows every inch of its bones, Jonathan Block.
This is a 50-million-year-old primate skeleton. It gives us a window straight into the world of what the earliest primates would have been doing, how they would have been interacting with their environment.
Wow. That's
the real
this is the real deal. That is absolutely exquisite.
Like most modern monkeys, Netharctus was a climber, adapted to life in the trees. The evolution of this creature, and others like it, had a huge impact on one of the features that most defines us, our hands.
One of the things that's really nice about this hand is that we do have all of the bones preserved.
It's like a jigsaw puzzle. You got to put it all together to see how that.
It's a
all fits.
three-dimensional
Yeah.
jigsaw puzzle.
To solve the puzzle of Netharctus's hands, you have to begin even further back in time. Over 365 million years ago, ancient fish used their fins to crawl out of the sea. Those fins evolved into the feet of reptiles, and later into the paws of mammals, with short fingers that all pointed the same way, and claws. Early primates like Netharctus took the mammalian hand to a whole new level, one that seems very familiar to us.
When you actually articulate all of these bones, what you see is that the thumb is divergent. That is, it forms an angle with the index finger. And so that shows you that it could bring its thumb into opposition with the rest of the digits.
Netharctus also has unusually long fingers and nails instead of claws. This is one of the first times in the fossil record that we see a hand that looks like our own, the hand of a primate. It's a turning point in the story of the human body, an anatomical change that would eventually let us shape the world around us.
다음엔 눈에 관련된 유산입니다. 원숭이는 정록 생명으로 적색과 녹색을 구별하지 못한다. 하지만 인간의 유전자를 이식하면 적색과 녹색을 구별할 수 있게 된다.
His work helped scientists figure out that most mammals, including most primates, see a limited range of colors.
This is Kramer.
Hi, Kramer.
And Kramer's red-green colorblind, but he has good blue-yellow color vision. In order to train them, we use the colors that they can see. So as you can see, that here is this yellow blob against a gray background. Kramer can see that as well as we can. If they get it right, they get a little reward, and they also get a clicking sound.
Oh, he was trying to kiss it there. He really did. He went, Mom.
You can see how good he is at it.
He is.
Kramer aces this first test.
You don't have any color vision, this is completely invisible. If you can't see red or green, this just looks like a totally gray background.
Most humans can clearly see this red blob, but Kramer can't. Like my son Nathaniel, he can't tell the difference between reds and greens.
Okay, Kramer, you were...
Rely on special proteins called opsins to detect color. They're held in thousands of special cells in the retina in the back of the eye. Kramer's got two types of opsin, each tuned to specific wavelengths of light. Signals from these opsins are then interpreted by the brain, which allows us to perceive color. But to see color like we do, Kramer would need a third opsin, tuned to different wavelengths of light. We think our early primate ancestors were like Kramer. They had just two opsins as well. So how did they evolve a third opsin? The answer is in our DNA. Each opsin is encoded by a single gene. And when scientists compared these genes, they found that the gene for the newer opsin sits right next to one of the old ones. And significantly, they are incredibly similar. Both facts are telltale clues as to how the extra gene evolved. The old opsin gene must have been duplicated. And one of these copies then acquired a small number of mutations that allowed it to detect different wavelengths of light. But there's one more question. Could our rich color vision result from just duplicating a gene? Or would there have to be changes to the brain as well?
It's actually a great evolutionary question. How did color vision evolve? How can something so complicated evolve?
Jay implanted a third opsin gene from a human directly into the retinas of a colorblind squirrel monkey called Sam.
What we did is really a test to see what's the minimal thing you could do in order to give an animal color vision.
The results were incredible. Like Kramer, Sam used to fail this test. Now he can easily tell the difference between reds and greens. Jay has recreated evolutionary history and given Sam human-like color vision.
You might think, oh, it would take them a long time to learn this new pattern in their brain. But as soon as the gene was turned on, the animal began to make these discriminations that they couldn't make before. The brain was already ready somehow. And so in one, very short evolutionary step, it goes to this totally different world. You go from just having strictly, let's say 5 colors, gray, black, white, blue, and yellow, to hundreds of different colors that are all the blues and greens and purples and oranges.
One simple shift opens the whole universe of color.
Yeah, that's the amazing thing. It's like there's something almost magical.
A dog has about 1,000 genes that are devoted to detecting odors. We have roughly the same number, but about 600 of them don't work anymore. They're relics. It's a similar story in other primates with color vision. These broken genes reveal another legacy of our primate past.
As our distant ancestors gained this wonderfully rich sense of color vision,
다음에는 두 발로 걷게 된 직립보행에 관련된 유산이죠.
Her bones are some of the best evidence we have for what early bipeds looked like.
Now, of course, if this were all articulated properly, with the vertebra on top of
You'd see
another,
it.
she'd be about 3.5 feet tall. You don't see that.
yeah, you don't get that.
Here's the femur.
That's the left.
top end of the thigh bone.
And this one also, I mean, when you put it all together, now you have, you know, this is telling bipedal, this is
Right.
giving a hint of bipedal. Bipedal.
This certainly tells it was bipedal. That can't in like that, which is very characteristic. Our knees are close together, and they
Yeah,
come
so-
up to the side. The chimps come straight up.
Having the upper and lower leg in a straight line is no good for a biped. It makes for an awkward waddling gait. Lucy's legs formed an angle. Her knees were closer together, just like our own knees. This positions the feet directly underneath the body, making walking easier and more efficient.
There we are. There we go.
So Lucy walked much like us. But she wasn't human. She had many primitive features too. Lucy tells us that by 3.2 million years ago, our ancestors had fully committed to walking on two legs. What she can't tell us is how our ancestors first started walking upright. To answer that question, we need to meet a second iconic fossil, found just 50 miles from the Lucy site. This fossil, called Ardipithecus, has turned our ideas about how we became bipedal on their head. The theory goes like this. We started out as apes that lived in the trees. We looked like chimpanzees and walked on all fours using the knuckles of our hands. But the climate changed. And only after forest had turned to savanna did we start walking on two legs instead of four. Artie tells a very different story. She was already walking upright while living in the woodlands. Anything anyone had seen before. It took a team of experts 15 years to fully piece her together. So how do we know she was bipedal? The man in charge of figuring out how Artie might have moved was anatomist Owen Lovejoy.
The critical thing in bipedality is the structure of the pelvis. And at first, when you look at this bone, you might think that there's not much information here. But actually, there's an enormous amount of information that tells you a great deal about what the original bone looked like.
Using information from this deformed bone, the team was able to reconstruct Artie's entire pelvis.
This is a plastic structure that's produced by a three-dimensional printer.
The top part of Artie's pelvis looks more human. The hip bone is short and broad, a key indicator of bipedalism. But the lower half is much longer than a human pelvis, more useful for climbing. The crucial time when our ancestors had just started walking on two legs. Artie was small, around 4 feet tall. A good climber, she moved on all fours when in the trees, but she walked upright when on the ground. She blows the old theory of how we became bipedal out of the water. Not only did our ancestors start walking on two legs when still living in the woods, they never looked or moved like our closest living relative, the chimpanzee. Some scientists don't accept this interpretation. They think Artie isn't a human ancestor at all, but the relic of an extinct ape.
다음은 뇌에 관련된 유산입니다. 대상 영속성이라는 용어가 있는데 존재하는 물체가 직접적으로 지각되지 않아도 지속적으로 존재하고 있음을 인식하는 능력을 말하는 거죠. 인간의 뇌는 다른 종에 비해 느리게 발달하는데 이는 단점이자 장점이 되는 거죠.
Monkey, ape, or human, our brains share a basic architecture that's different from most other mammals. We all have a special region involved with hand-eye coordination, and a greatly expanded visual system with as much as 40% of the brain involved in seeing. Our inner monkey hasn't just shaped our bodies, it's shaped our brains as well.
You're going to put a piece of fruit on the toy itself. You'll place it in the well,
She's looking at it. There.
and you'll move that to completely hide that toy.
Okay.
There you go.
Wow.
There you go.
Wow. That is impressive.
Object
Just
permanence.
boom.
3 months old, and already CI understands object permanence.
CI, you are good.
Hi, Geneva. How are you? You are a cutie.
Geneva is the same age as the baby monkey. I'm going to do the exact same test on her under the expert eye of child development specialist, Professor Susan Speaker.
She needs to reach for him, show that she's interested.
Oh, I think we got interest.
She's got a good reach.
You do. Look at you. Oh, Geneva, you like those. Hello.
Now, Neil, can you get them away?
Oh, I'm an expert at this. Can I take that for a second? Sorry, be temporary. Yeah, you are so good that way. I'll pop it in. And
Make sure she's watching.
She's watching. Now we'll cover it up. Watch this.
And she wonders, what did you do, Neil?
What toys?
I don't know where they went.
Wow, that is amazing.
Where did they go?
It is incredible. If it's not there, it doesn't exist. That is amazing.
So how come a baby monkey beats a baby human in this test? The brain of a fish and the brain of a human have more in common than you might think. This is a trip to the beginning. This is as basic as it gets. We're going human fish, represented by a cartilaginous fish, a shark on the right. And what you have is you see the muscles and nerves and organs inside the shark head. But importantly, you see this yellow tissue. This is the front end of the spinal cord. It's the brain. Doesn't look like much, but it's the shark brain. This is the human brain. This is where it all happens for us, where our memories reside, where our fears, our loves, our hates. It's all in here. The human brain, really at the surface level, looks nothing like what's sitting inside the shark head. Yet when you know how to look, you find a very profound similarity. And that similarity lies in the overall architecture of the brain itself. In the human, we have a forebrain. And that forebrain consists of this folded tissue here, where much of our conscious thought happens. And then there's a midbrain, this walnut-sized area in the middle. And then a hindbrain. That's a fundamental division. We see that in development, and we see that based on where the nerves exit the brain. Well, if you follow the similar nerves in a shark, what you see is sharks too have that same structure. A forebrain, a midbrain, and a hindbrain. There's a fundamental architectural similarity among all brains. The brains of people, the brains of dogs, the brains of cats, the brains of monkeys, lizards, frogs, salamanders, trout, bass, even sharks and fish are similar. Each one of them has a forebrain and a midbrain and a hindbrain, despite the fact that those brains are often very different in function and form.
다음에는 창고기의 뇌 DNA와 비교하는 겁니다. 신경사기라는 말은 원시적인 신경관으로서 뇌나 척수로 발달하는 부분을 말하는 거죠.
Deep in the genes of this animal and the development of this animal, and deep in the genes of us and the development of us, there are the similarities.
They're pretty active.
They're pretty active.
Holy cow. Oh, that is beautiful. God, they're so clear too. You can see right through them. It's amazing.
Amphioxus lives in the sand of the ocean floor, filtering algae out of the water. It's from an incredibly ancient group of animals. We found fossils that look a lot like this in rocks that are over 500 million years old. They are a window into what came before the first fish. And crucially, they don't have anything like what we'd call a brain. The simple nerve cord that runs the length of their body ends with just a tiny swelling invisible to the naked eye.
If you look at the front end of this animal, you don't see it all expanded into a large skull or brain region. It's just pointed.
But if you look at the genetic makeup of these ancient creatures, you find something truly remarkable. To make the forebrain, midbrain, and hindbrain that we saw in our lab, you need a series of control genes to tell the cells within the developing brain where they are. they're like a zip code. Those same control genes that make our own brains are active in amphioxus. But here, they're simply building the primitive nerve cord and the tiny swelling at its front. This means that the genetic roots of our complex primate brains existed in much simpler creatures that first arose over 500 million years ago.
I think it gives us a glimpse into where our brains came from, into the basic organization of the brain of our ancestors.
We've seen how our distant relatives, from worms to fish, from reptiles to early primates, have defined our bodies. At each new stage of our journey along the tree of life, our ancient animal ancestors have reconfigured what's gone before. And that's how we ended up as the intelligent creatures with all our quirky flaws that dominate the planet today.
There's something incredibly profound, and I think beautiful, in the idea that inside every organ, cell, and gene of our body lie deep connections to the rest of life on our planet.